what we're a background on what we're doing and why we're doing it. And then I'm going to talk about the NSF transcriptome experiment that we performed, which is just one part of this plant genome grant, some of our results from that. And then I'm going to go on to talk about some functional experimentation on one of the candidates we identified. Okay. So I won't belabor this too much because I'm sure you all already are very well aware. But annual yield loss in cacao is caused by a variety of pathogens. Here, I'm showing you three of some of sort of the big baddies. On the left-hand side, we have frosty pod rot. On the right-hand side, we have witch's broom. And in the center, we have black pod rot. And this last one, black pod rot, is the worst of these pathogens and is caused by 4 separate Phytophthora species. I'm going to be focusing on, and actually the broader NSF grant that Mark mentioned focuses on Phytophthora palmivora. So whenever I say black pod rot or Phytophthora from here on out, I'm just exclusively referring to palmivora. Okay. And while there are clones that have been generated that have resistance to Phytophthora palmivora or black pod rot generally, they come from a narrow set of parents. And so they're not necessarily representative of cacao's broader genetic diversity. So the goal of the NSF project was to identify and characterize genes that are involved in resistance to Phytophthora palmivora through a number of different technologies, including whole genome and transcriptome sequencing, as well as some metabolite analysis as well. And we wanted to identify genes that are involved in resistance across a diverse set of cacao populations. And there are a few different questions that we're sort of seeking to address here, but I'm just going to be talking about these two today, which is what genes respond to pathogen challenge both within and across the populations that we looked at? And then are there sort of core functional groups or pathways or genes that are involved or critical for resistance? So to start this, we assayed genotypes from a number of populations across cacao's native range. We're again looking at resistance to Phytophthora palmivora. Here, the populations that we looked at were Guyana, Iquitos, Marañón, and Nanay. And here I'm showing you their presumed centers of origin. And so the phenotyping was done by a former postdoc in the lab, Drew Pfister, as well as a researcher at Cartier, Mariela Leandro Munoz. And what they did was they took leaves from cacao genotypes. They performed what are called detached leaf assays, where they cut the leaf, they put it in a petri dish, they challenged one side with pathogen, in this case again palmivora, and on the right-hand side they challenged it with control or just V8 media plugs. They came back 48 hours later and they looked at lesion growth. The top part is showing, the top line here that I'm highlighting is showing you what a susceptible genotype would look like, where we see lesions that have greatly expanded over the course of 48 hours, sometimes even merging. And on the bottom, I'm showing you what resistant genotypes would look like, where there is a visible necrosis, but not nearly as bad as the susceptible genotypes. And so when we look at all 70 or so genotypes that were investigated, what we see is a gradation from highly susceptible to highly resistant. So here, the y-axis is individual genotype, the x-axis is average lesion radius, the top then represents genotypes that are highly susceptible, and the bottom represents genotypes that are highly resistant. And as we can see, there are both genotypes that are considered resistant and genotypes that are considered susceptible within each of these 4 populations. So we took the 4 most resistant and the 4 most susceptible genotypes from each of these populations. We imported the grafted plants from Costa Rica to Penn State. And from those grafted plants, we took rooted cuttings for our transcriptome experiment. We took about 300 and ended up with about 160 that survived. We took those approximately 160 rooted cuttings and we distributed them across the greenhouse in sort of a randomized block design, where we had, that's what I'm showing you here on the left. We had 2 separate tables. Each table had 3 trays, alternating pathogen treatment or control. And what we tried to do was take genotype, we would mirror genotype, the position of genotypes across tables. So if there was a genotype here shown in purple, we'd try to mirror its position on the other table. Okay, so we went through, after they were distributed across these tables, we went through and challenged these leaves with pathogen. On the underside of the leaf, we would place mycelial plugs. And then 8 hours later, we came back, we collected tissue from surrounding areas surrounding that lesion, and we carried them forward for RNA sequencing. Okay, so the whole workflow would look something like this, where we import the plants from Costa Rica, we set up the design, we do the experiment, we perform RNA extraction and sequencing. We do a number of pre-processing steps, which actually involved some de novo assembly of quite a few genomes, which I won't get into today, but if you want to see some other work that's come out of the project, there are a number of population genetic papers. And then we performed a series of analyses, differential expression analyses. And this work was done by myself as in partnership with a former technician in Mark Anciala's lab named Melanie Perryman, as well as one of our key collaborators, Eric Wafula, who's a bioinformatics scientist. And we were primarily interested in differentially expressed genes that were behaving in sort of three ways. The first is that are genes that are responding to pathogen treatment. So those would be genes that look something like this, where y-axis is expression, and then x-axis is control versus treatment. So a gene would increase in response to pathogen treatment, but there's no difference between resistant or susceptible phenotype. That's shown by the color. We were also interested in genes that are responding to resistance versus susceptible phenotypes. So again, y-axis is expression, and this, what we would see here is a gene that there's a baseline difference in expression between resistant and susceptible, but there's no response to control or treatment. And lastly, we were also interested in genes that have sort of an additive effect, which is they have a baseline difference between R versus S, but they are not, but they are also responding to control and treatment. So that's what we're calling additive. And so if you remember back, that first question that we were interested in is what genes are responding both within and across populations. So we began by visualizing that using what are called upset plots, which I know are a little intimidating, but actually I think end up being more intuitive, so bear with me. So here, the colored bars show you differentially expressed genes that are responding in only a single population. So here, red, we see that there are 429 genes that are just responding in Iquitos. That would be similar to a Venn diagram that looks something like this. Blue represents 417 genes that are only responding in Guyana and not shared across the other genotypes, et cetera. And then when we get to black bars, what we see are genes that are shared across genetic groups. So here, 119 genes are shared across Guyana and Nene. And by the time we get to the far right, what we see are genes that are shared, differentially expressed across all four genetic groups. Okay. So what we see when we then investigate those three main effects that I was talking about, treatment, phenotype, and additive, is that many of the genes responding in each population are unique. So here on the far left, we see about 40% of the genes that are differentially expressed across these four genetic groups are only found within those genetic groups. And there's actually, then the rest of this, only 60% is shared across all of them, or shared across some combination of all four. And we see that pattern for resistant/susceptible phenotype as well as additive phenotype, or additive effect as well. And secondly, what we see is that log fold change is only weakly correlated across these genetic groups. So if we took a single gene and we looked at how it was behaving across all four groups, it's likely that it wouldn't be doing the same thing. So it appears that these cacao genetic groups are responding differently to either pathogen treatment or resistant/susceptible phenotype, that they're using largely different sets of genes to be resistant. Okay. However, among the sets of differentially expressed genes that are shared across populations, we see sort of a cast of usual suspects. So for here, for instance, we see a number of worky transcription factors that are well-known mediators of plant defense response, as well as several MIB transcription factors that are known to be involved in secondary metabolite accumulation, as well as chitinase and peroxidase, pathogenesis-related genes, and then a number of genes in the phenylpropanoid pathway. And it's these phenylpropanoid genes that I'm going to be focusing on for the second part of the talk, which is looking at some functional characterization. And these phenylpropanoid genes were immediately interesting to us for a number of reasons. First, they're well-known phenylpropanoids and polyphenolics that come from that pathway are well-known mediators of plant defense response, including metacarpin and resveratrol, as well as many others. But also they actually have a demonstrated role in the cacao Palmivora interaction. So Ben Nolenberg, a former postdoc in Mark and Ciela's lab, did some work where he discovered this compound or characterized this compound clovamide, which is a phenylpropanoid compound, and found that it significantly inhibited 3 different species of Phytophthora when the plates were dosed with clovamide. And we're also interested in this particular pathway because there's a number of the genes within it are responding consistently across all 4 populations. So one of those genes, the ones I'm going to focus on, is caffeoyl-shikimate esterase, which on the left-hand side, what I'm showing you is the y-axis, which is normalized expression, and then on the x-axis, which is control versus treatment for each of the 4 populations. And what we see is that this particular gene, CSE, is consistently upregulated across all 4 populations in response to pathogen, suggesting it's potentially critical for pathogen defense. And then secondly, CSE leads to caffeic acid accumulation. I'm showing you here, CSE is highlighted by that red circle, and it catalyzes this reaction from caffeoyl-shikimate to caffeic acid. And caffeic acid is interesting because it has been characterized to be, it has been shown to inhibit zoospore accumulation or germination for both Phytophthora palmivora and Phytophthora megakarya. So it's immediately sort of an interesting target. So with that in mind, Ben and I set out to perform several experiments to characterize CSE and its importance in this cacao Palmivora interaction. We first we cloned and overexpressed TCCSE in Benthamiyana and looked at accumulation of caffeic acid 48 and 96 hours post-transformation. Here, the y-axis is showing you relative abundance of caffeic acid, and then on the x-axis, I'm showing you overexpression of TCCSE versus the empty vector control at both 48 and 96 hours. And what we see is that at both 48 and 96 hours, the plants that are overexpressing the CSE gene accumulate caffeic acid. Okay, this is consistent with what we would expect its function to be. Secondly, we performed 2 experiments to show that caffeic acid is directly inhibitory to Phytophthora. We, like I showed before, that caffeic acid inhibits zoospore germination, but it was unknown whether or not it would inhibit mycelia as well. So we performed an experiment where we dosed cultures of V8 media with 2 millimolar caffeic acid, and we looked at inhibition of Phytophthora palmivora. So here the y-axis is area, and then the x-axis is either V8 control or 2 millimolar caffeic acid. And what we see is significant inhibition of Phytophthora palmivora when we treat it with caffeic acid. And secondly, Ben came up with this creative experiment to test caffeic acid mobilization to the site of infection by, and what he did is he took leaves, and on the underside of leaves, he put either a droplet of water or a droplet of zoospore, came back 24 hours later, sucked up those droplets and put them through LCMS to look at caffeic acid accumulation. And what he found is that, again here, y-axis is relative abundance. And then x-axis represents either water plus leaf, zoospore plus leaf, or zoospore only. And what he found after 24 hours was that the leaves that were treated with zoospores, those droplets significantly accumulated caffeic acid, consistent with this caffeic acid being mobilized to the site of infection. So it stands to reason then that caffeic acid should accumulate in pathogen challenged tissue. So what we did is we went back to our tissue from the transcriptome experiment. We extracted metabolites. We extracted metabolites from that tissue, and we put it through mass spec. Here, y-axis again shows relative abundance. Blue versus yellow represents control versus treatment. And what we expected to see was actually caffeic acid to increase. However, we saw a decrease in the pathogen treated tissue, albeit not significantly. So it's a little bit counterintuitive response. So where is the caffeic acid going? It could be going in a number of different directions, and we're trying to figure this out. One possible explanation is that caffeic acid is derivatized into many different caffeic acid derivatives that are known to be antimicrobial, but would be hard to track down and quantify. Another possibility is that caffeic acid could be shunted into lignin, which is insoluble in the protocol that the metabolic extraction protocol that we used, and so we wouldn't have actually seen that. And then a last possibility is time point. So most of our experiments were done after 24 hours at 24, 48, 96. So it might be the case that our 8-hour time point for the transcriptome experiment is just a bit too early. So in conclusion, different sets of genes respond to Phytophthora palmivora across the 4 genetic groups that we investigated, but there are core functional pathways that are shared between populations. So there are a number of genes that are differentially expressed in this core phenylpropanoid pathway, and I showed you results for some characterization of just one of them, caffeoyl-shikimate esterase. Overexpression of this gene results in increased caffeic acid accumulation in tobacco, and it also is, we showed that caffeic acid significantly inhibits mycelia and is mobilized to the site of pathogen infection. However, cacao challenged palmivora, cacao challenged with Phytophthora palmivora does not appear to accumulate caffeic acid, a little bit counterintuitively, but there are some potential explanations. Okay. And with that, I would like to thank the broader NSF group, as well as the other various lab groups that I am a part of. I tried to highlight people who worked on what I showed today. One potential person that I missed, though, is Naomi Altman, who was really integral to helping us with our transcriptome experimental design and analysis. And with that, I would be happy to take any questions. Thank you.
Thanks, Noah. We have about 3 or 4 minutes. We could probably take a couple of questions.
Noah, this is Brian Bailey down in Beltsville.
Mm-hmm.
When you were looking at the accumulation of the caffeic acid in response to the zoospores infection, what other compounds were with the caffeic acid?
Yeah, so we used LCMS, like I said, so we have the potential to look at accumulation of other metabolites, but we were primarily focused on caffeic acid for now. So we haven't looked a ton at the other compounds that are accumulating. However, we do see, from what we have looked at, we do see common things like flavonoids. We see a lot of flavonoid accumulation. But yeah, that we need to do some more work on figuring out what else is accumulating.
So do you think it's a specific release, or is it just a general leakage of metabolites?
I would say that it is a specific release because we see there are metabolites that are accumulating specifically in our disease challenged tissue. We just haven't gone through the work of characterizing necessarily what those are, which indicates that they're responsive to the pathogen. If that answers your question.
That's close. Thanks, Sam.